Biology 1108, Presentation Number 2, Evolution. In this presentation, we will begin to start focusing on Darwinian evolution and this mechanism we call natural selection. We will review some of the information that we covered in presentation number 1. That will reinforce that information, and we will in turn then apply it to our current knowledge of what constitutes natural selection and what constitutes Darwinian evolution. Okay, so let's get started. Evolution is descent with modification. That's an observation we made in presentation number 1. Evolution is change. Evolutionary change means within a species we are examining changes in gene frequencies, and those changes in gene frequencies affect the overall population. So ultimately the species does change over time. And it's not without some overriding reason. The fact of the matter is any of this change and these modifications, or what we might officially refer to as adaptations, will actually improve the fitness of the species. And so I will remind you, and we will cover this again and again and again, fitness is a combination of two things: the ability to survive and the ability to reproduce. If you survive, your genes are preserved. If you reproduce, they are preserved in the sense that they're able to be passed on from generation to generation to generation. So it's a consequence. That cactus plant that I see on the slide demonstrates a number of adaptations. Some of the most obvious adaptations are the fact that what would be leaves are spines that protects the cactus plant from being eaten, and the cactus plant has this big, thick, fat, fleshy stem that allows it to store water. Your ability to store water, your ability to prevent yourself from being eaten, both of those will be a huge contribution to your fitness, your ability to survive, and your ability to reproduce. So adaptations are evidenced as variation in plants, in animals. A lot of the variety we see are modifications that improve the ability of that animal, of that plant to survive and reproduce and pass those genes on to future generations. Now, the reason why I emphasize the word those genes is because those genes are what actually are responsible for encoding those adaptations. The cactus plant's spines, the cactus plant's ability to store water, the animal's claws, the animal's coloration, the animal's behavior. Now, you'll remember that these adaptations are collectively referred to as phenotype. Phenotype, you'll remember, is the expression of one's genes. And so your genes can be expressed as variations in morphology, what you look like, variations in physiology, how you operate, and variations in behavior. And all of those conceivably contribute to your ability to survive and reproduce in the environment in which you live. So all this variation, these adaptations we've been talking about, technically these are the raw materials for natural selection, because natural selection determines which of those genes, which of those characteristics are selected against or selected for. If they're selected against, they won't become established in the species, but if they're selected for, well, then the species will essentially demonstrate those characteristics. Taking a look at the illustration on the slide, which is vaguely reminiscent of an illustration we saw earlier before, I see a bunch of ducks. And I see I'm selecting against the lighter colored ducks, which means ultimately my species will be predominantly darker colored ducks because I am both selecting against the lighter colored ducks, which means in turn I'm selecting for the darker colored ducks, and I'm getting a population that was largely much more fit, much more able to survive, much more able to reproduce than I had previously. Not really necessary to focus a long time on this particular slide. Basically reinforces what we already know. Natural selection is a mechanism by which we select gene combinations that are most fit. By selecting gene combinations that are most fit, we're technically selecting for phenotypes that are most fit. So as a mechanism, natural selection really is a filter. It selects for or against certain characteristics, determining whether those characteristics will ultimately be passed on to future generations or not. To borrow some terms from a previous lecture on genetics way back in a previous course, the word genotype means the alleles or the specific genes you have, and how those genes are expressed is phenotype. So really, genotype is what natural selection is allowing to pass forward from generation to generation. But ultimately, phenotype is where the rubber hits the road, because that's the point of where those genes determine whether you get to survive or reproduce. So variation is encoded in genotype in the form of genes or specific states of those genes, which we called alleles once upon a time, and is ultimately manifested as phenotype, more specifically variations in morphology, variations in physiology, and variations in behavior. So if variation is the raw material for natural selection, where does that variation come from? Let's identify some important sources of variation, and the most important source of variation is where genes come from. Any new gene has to arise from some change in a gene sequence, and that's called a mutation. A mutation is any change in a gene sequence, and it is the ultimate source of new alleles. All I have to do is change an A, C, T, or G, and that could conceivably change what that gene codes for. And if I change what the gene codes for, well, that could give me a beneficial gene, or it could be one that's deleterious, or it might be one that has no effect at all. But ultimately, I need you to know that mutation is the ultimate source of new alleles in any particular species. And in order for that allele or that gene to become integrated into the genome, to become part of that species, it must be selected for. Now, there's another way to think of how variation might arise in a species. And that's because many of the species we'll be talking about are sexually reproducing species. Animals, plants, fungi, many of the protists are sexually reproductive. Now, in doing sexual reproduction, you understand at its most basic, you've got a parent contributing one set of alleles. You've got a second parent contributing another set of alleles, which means the offspring is a unique combination of the two. One could then surmise that the offspring has its own particular characteristics that might promote its fitness or might not promote its fitness. So essentially the variation found in the offspring is a consequence of the inherited genes coming from parent A and parent B. That's not all that's involved here, though. So remember that sexual reproduction means the formation of gametes or sex cells. And the formation of gametes or sex cells in a parent will involve this process called meiosis. And in meiosis, there is this thing called crossing over. You remember that's where you've got homologous chromosomes exchanging bits and pieces. It's like shuffling genes between those chromosomes. And while that seems very, very, very small and very, very highly focused, ultimately that change, that variation, is important in terms of the organism that receives those new combinations. Then, through the process of meiosis, you get independent assortment, which means your gametes will ultimately be receiving those genes. And so, once again, you're shuffling the genes to determine which one's actually going to go into a new offspring. And then finally, the syngamy between different mating genotypes, that is, fertilization between any number of different sperm types and any different egg types, are going to come up with unique zygotes. So ultimately, in any species, mutation is your ultimate source of new alleles, but then again, got to understand there are different things going on in the sexual reproduction process, crossing over during meiosis, independent assortment during meiosis, and the syngamy of different mating genotypes, giving me really, really unique offspring. And this is the raw material, what natural selection is ultimately going to act on. We've already made the observation that individuals do not evolve. Natural selection does not act on individuals, even though individuals are an important component of what natural selection actually does. But the importance of the population cannot be underscored, because the population is our basic unit of evolution. The population is where we find these genes. And you remember, genes come in different types. They call those alleles. And so if you've got a population, a group of interbreeding individuals, and they are all interbreeding and sharing these genes, those genes constitute what is called a gene pool, and its natural selection is having an effect on allele frequencies in that population. You take a look at the wild pigs on the right, and you realize that they are full of different alleles, big B's, little b's. We're being as simplistic as possible, but the idea, it's the process of reproduction in those pigs that's making certain those big B's and those little b's get spread from the parental generations to the offspring generations. But then ultimately, it's natural selection which is going to influence the survivability of the reproducibility of those pigs, will determine how many of those big B's and how many of those little b's actually get passed on to future generations. And that sense of changing allele frequencies over time is what evolution actually is. All right, well, let's take a look at allele frequencies. Now, allele frequencies are the percentage of locations in a population that will be occupied by a given allele. Do I find 90% of the individuals in the population have a given allele, or only 10%? And so that's what allele frequencies are all about. We already made the observation that natural selection is going to influence those allele frequencies over time. Now, if allele frequencies change within a population, we're going to call that microevolution. But now, in the process of watching how these allele frequencies change over time, at any given moment, those allele frequencies are going to be in some stable situation. For example, take a look at our ducks over there on the right. Count down to the fourth generation, and at the fourth generation you will see a series of ducks, a couple of lighter colored ducks, a middle colored duck, and a really, really dark colored duck. At that moment in time, even though I know in my heart of hearts that the allele frequencies are changing because of selective pressures against the lighter colored ducks and selective pressures for the darker colored ducks, at that instant in time, I see what looks like 2 out of 4 ducks in the population are lighter colored, 1 out of 4 ducks in the population is middle colored, and 1 out of 4 ducks is darker colored. In other words, the allele frequencies at that instant of time, 25% dark ducks, 25% intermediate ducks, and 50% lighter colored ducks in the population. Now, that is described as a Hardy-Weinberg equilibrium at any given instant in time. Even though I'm understanding that this population is changing over time, if I took a camera and took a genetic snapshot of that population at that point, my population's genes are in equilibrium. Now, we're going to talk more about this on the next couple of slides. It's called the Hardy-Weinberg equilibrium. So let's take a look at what this might look like if we graph it. If I take a look at a graph of allele frequencies, I see here that the frequency of the dominant allele A, as time goes by, and the same is true for the recessive allele A, indicated by the red line. And so I see within the population there is change going on over time. So I've got time illustrated by these arbitrary numbers down below, but the number 6 would imply maybe over 6 generations. I see these alleles varying in frequency. Now, at any given point in that sequence of time, those alleles are going to be at some form of equilibrium. And let's take a look at our next slide and we'll see that. So here's our next slide. And we see the big arrow indicating the evolution of population X over time. I see the changing gene frequencies, but at the instant point, which corresponds with the number 5 on the x-axis, at that point, I could actually compute exactly how many individuals in the population have the recessive allele, how many individuals have the dominant allele, and not only that, I should be able to compute how many of those individuals are big A, homozygous dominant, how many of those individuals might be little a, homozygous recessive, and how many might actually be heterozygous, big A, little a. In other words, there should be some way that I could actually mathematically compute all that information just from one quick snapshot of what's going on in that population at an instant of time. So this thing that we're calling the Hardy-Weinberg equilibrium is the idea that at that one instant in time, I have an ability, a mathematical way to determine the frequencies of genes in a population at an instant of time. And we're going to take a look at this in detail in the next slide or two. The details of the Hardy-Weinberg are going to cause us to have to go back and review some things that we know from a previous presentation, maybe long ago and far away, where we learned about alleles and we learned about genetics. So here is our review. Alleles can be dominant, uppercase A, or they can be recessive, lowercase a. I could ask the question, what percentage of loci or gene locations in the population are occupied by the dominant allele or the recessive allele? At that point in time, how many big A's are there and how many little a's are there? And then on my next bullet point, I will remind myself individuals are a combination of two different alleles. You inherit one from one parent and you inherit the other from the other parent, which means you could have two dominant alleles. You could be homozygous dominant. You could have two recessive alleles. You could be homozygous recessive, or you could be heterozygous, having inherited a dominant allele from one parent and a recessive from the other. If I know how many loci are occupied by the dominant A and how many are occupied recessive A, I should be able to compute the percentage of individuals in the population who might be homozygous dominant, who might be homozygous recessive, or who might be heterozygous. So we take what we know about genetics. We can actually turn these letters, these P's, these Q's, these homozygous dominants, homozygous recessives, and heterozygous, we can turn these into variables that we can actually plug into an equation. You will need to know these variables. They are found on this slide. They're actually going to be found on the next slide as well. So here's what we need to know. The letter P corresponds to the frequency of the dominant allele in the population. If 75% of the loci in the population is occupied by the dominant allele, then P is 0.75. If 50% of the loci in the population are occupied by the dominant allele, then P is 0.5. If 25% of the loci in the population is occupied by the dominant allele, well, then P would be 0.25. Q, on the other hand, is the frequency of the recessive allele in the population, and likewise, if 25% of the loci in the population is occupied by that recessive allele, well, then Q is going to be 0.25. If 50% of the loci in the population is occupied by the recessive allele, well, then Q is going to be 0.5. Well, now these expressions, P and Q, can be used as a starting point for generating expressions that will allow us to figure out the frequency of homozygous dominance in the population, the frequency of homozygous recessives in the population, and the frequency of heterozygous individuals in the population. You will notice under the bullet point genotype frequencies on the particular slide, I've designated big A as the dominant allele, little a as the recessive allele, and you will see over there on the left the expressions that correspond to that. If P is the frequency of the dominant allele in the population, well then P squared, or P times P, is going to be the frequency of homozygous dominance in the population. If the frequency of the dominant allele in the population is 0.5, that means P squared is 0.25. Similarly, Q squared. Q times Q, the frequency of homozygous recessives in the population. And then what you got left over is going to be the frequency of heterozygous individuals in the population, individuals that are big A, little a, and that expression is 2 times P times Q. There are going to be two equations that are critical to our ability to perform any Hardy-Weinberg calculation. The two equations are P plus Q equals 1, and the second equation is P squared plus 2PQ plus Q squared equals 1. Let's take a look at the two of these. The first equation, P plus Q equals 1, is relatively simple. It is where the letter P corresponds to the frequency of the dominant allele in the population. Q corresponds to the frequency of the recessive allele in the population. Let's suppose 50% of the loci in a population is occupied by the dominant allele. 50% is 0.5. So P would be 0.5. That would mean that the other 50% of the loci are occupied by the recessive allele, and that would be Q. So 0.5 for P plus 0.5 for Q equals 1, or 100% of the population. So the equation P plus Q is going to be used to determine the overall percentage of the dominant allele loci or recessive allele loci found in the population, but it's also going to be critical for our ability to figure out what P or Q actually is when we start thinking about this second equation. So our second equation here is going to help us to resolve exactly what proportion of the population are homozygous dominant individuals, what proportion of the population are homozygous recessive individuals, and what portion of the population consists of heterozygous individuals. The expression P squared is the frequency of homozygous dominant genotypes in the population. The expression 2PQ is the frequency of heterozygous individuals in the population. And then finally, the expression Q squared corresponds to the frequency of homozygous recessives in the population. So let's think about a gene A. The gene A comes in two different forms: a dominant, which is the uppercase A, and the recessive, which is the lowercase a. Like I said, if 50% of the population is big A and 50% of the population is little a, that is explained by our first equation there. Then if I took the 0.5 and I multiplied it by itself, that's 0.5 squared, that's P squared, that would give me the overall percentage in the population, which are big A, big A. So 0.5 squared is 0.25, which is 25%. So we turn our attention to the Q squared expression, be the same thing. Q squared is going to be 0.5 squared, which will also give me 25% of the population. Let's take a look at the expression 2PQ. Exactly what proportion of the population are going to be heterozygous individuals? Well, that would be 2 times 0.5 times 0.5. which turns out to be 0.5. So I'm looking at this second equation. 25% of the individuals are going to be homozygous dominant, 25% of the individuals are going to be homozygous recessive, and 50% of the individuals are going to be heterozygous. So very easily I can compute any one of these variables from the information given to me in any of these equations. Now, Hardy-Weinberg is a model. A model is a representation of reality. Models necessarily are simplified versions of reality. It's a way that we're able to actually describe reality in a simplistic manner. So Hardy-Weinberg is a model, and we are going to realize that models are not perfectly realistic. In terms of the Hardy-Weinberg equilibrium, there's a number of assumptions we're going to need to make. Any violations of these assumptions really makes Hardy-Weinberg impossible. So here's the assumptions. And they're not terribly unreasonable, but the fact of the matter is they're not necessarily realistic. So we're going to begin with the Hardy-Weinberg equilibrium assumption number one is we're talking about an extremely large population. We're not talking about 1,000 individuals. We're not talking about 10,000 individuals. We're talking about millions of individuals. The larger the population, the less opportunity there is for random variations. Hardy-Weinberg assumption number two, our population is a closed population. A closed population sees no new alleles coming in, and there are no alleles going out. Anything that's affecting the change in allele frequency in the population is within the population itself. We can't have any outside influences. Hardy-Weinberg assumption number three, no mutation. Obviously, a mutation is going to create a change. If you have a change, that's going to mess up the equilibrium. So technically, you can't have mutation going on in Hardy-Weinberg. Random mating is the idea that any given individual in the population has the capacity to mate with any other individual. And that's going to ensure completely and total random distribution of alleles in the population. And then finally, Hardy-Weinberg assumption number five here, no natural selection. Natural selection changes gene frequencies. We can't have that happening. Because you understand, Hardy-Weinberg is a way of measuring the relative number of alleles in a population at an instant of time. Anything that's going to mess that up is going to mess up the equilibrium. So hence, Hardy-Weinberg, like I said, is a model. It is a mathematical snapshot of how many alleles there are and how many individuals have those alleles in specific combinations. But anything that's going to have an influence on those alleles and their frequencies in the population is going to be something that is going to violate the model, and so therefore we make these assumptions. And the understanding, of course, is that you'd have to kind of hold your breath when you were applying Hardy-Weinberg to a small population. You'd have to hold your breath when you apply Hardy-Weinberg to a population experiencing immigration and emigration. And you certainly couldn't apply Hardy-Weinberg to a population where the individuals are performing non-random mating. They're choosing mates instead of randomly mating with each other. Well, even if those assumptions seem terribly unrealistic, it doesn't change the fact that Hardy-Weinberg is a pretty darn effective way of predicting allele frequencies or genotype frequencies in a population. So what we're going to do is going to take a look at a real life example. So let's read the question. I'm going to have to provide a little bit of explanation towards this. If 9% of an African population of humans is born with a severe form of sickle cell anemia, sickle cell anemia is represented by the genotype little s, homozygous recessive, what percentage of the population will be more resistant to malaria because they are heterozygous, big S, little s, for the sickle cell gene? Now here's the additional explanation. Sickle cell anemia will kill you. If you inherit a recessive allele from your mom and a recessive allele from your dad, you will have sickle cell anemia and you will more than likely die. Now, on the other hand, it turns out that if you inherit just one gene for sickle cell anemia, if you are heterozygous, you got one gene for sickle cell anemia from one parent, but a normal gene, S, from the other parent. If you are heterozygous, you aren't going to get sickle cell anemia. And the added benefit, though, is that if you have that sickle cell gene there, it makes it really, really difficult for the malaria parasite to become established in your red blood cells. So malaria, which is a really, really critical public health issue in Africa, is not going to be a problem for you if you've inherited one copy of the sickle cell gene. So obviously from a public standpoint, a public health standpoint, this question makes sense. If 9% of an African population is born with a severe form of sickle cell anemia, what percentage of the population might be more resistant to malaria because they have inherited the heterozygous genotype for the sickle cell gene? In other words, what percentage of this population is going to be resistant? So let's take a look at the process and the answer. Now remember, we're using these variables and we're using these equations to figure this out. See, we're given some basic information in the question. We're understanding that 9% of the African population is born with a little s genotype. Now, you and I both know that the little s genotype is Q squared. So we could take the square root of 9%, that would give us Q. So if Q squared is 9% or 0.09, it's the number of individuals in the population of little s, let's do a square root. The square root of 0.09 is 0.3. That's Q. Once we get Q, we can solve for P. If I remember correctly, P plus Q equals 1, which means P plus 0.3 equals 1, which means that P is 0.7. So we already have solved for Q, and we've already solved for P. Now we need to figure out how many individuals in the population are heterozygous big S, little s. Well, we already know that that particular expression in the equation is 2PQ. So all we have to do is plug them in. 2 multiplied by P, which is 0.7, multiplied by Q, which is 0.3, gives me 0.42, which means 42% of this population are heterozygous, which means 42% of this population are going to be resistant to malaria, even though 9% of the population is being born with sickle cell anemia, and that's a problem. But the silver lining, I guess you could say, is even though 9% are facing mortality due to sickle cell anemia, a full 42% of the population are looking at potential protection from a very dangerous disease, malaria. So now we're about to head for an explanation of the evidence for natural selection. But before we do that, I apologize. I'm going to flog a dead horse. We're going to cover natural selection yet one more time. And I really hope, it's going to set in. So that's one of the reasons why we're hitting it once more in 2 successive slides. Remember, natural selection is the mechanism of evolution. It's how nature selects the gene combinations are most fit. It's not the case where nature consciously does this, but the fact of the matter is nature functions like a filter between one generation and the next, and if you are fit, You get to survive, you get to reproduce, you pass your genes on to future generations. If you are not fit, you don't survive, you don't reproduce, you don't pass those genes. As a consequence, the genes might even be lost from the population. So the basic idea is, with this filtering process going on, gene frequencies in the population are going to change over time. So if I've got changes in genes, or more specifically changes in alleles, that's going to change genotypic frequencies. That is the genetic composition of the population. And that can also influence phenotypic frequencies. And you remember, phenotype is the expression of those genes. So if we were talking about rabbits, changes in genotypic frequencies could ultimately change phenotypic frequencies, in the sense that a rabbit population might see the loss of lighter colored rabbits and the predominance of darker colored rabbits because of that process of natural selection. So what about this evidence for natural selection? We're about to look at an actual historical bit of work done by a fellow named Kettlewell in the 1950s. And Kettlewell, who you see is pictured down there, was an ecologist, and he was an entomologist, and he was interested in moths. Of all moths in particular, he was interested in the common peppered moth, which is a fairly common indigenous moth found in England. Now, England is a place where they've got collections of things in museums, and some of the collections they got are really, really old. So Kettlewell bounced around in museums of England looking at collections of the common peppered moth, and he discovered that in collections made before the industrialization of England, which happened sometime during the 18th century, the 1700s, the common peppered moths in those collections were lighter in color. After the Industrial Revolution, after the 1700s, the common peppered moths happened to be darker in color. Now, he knew that these were still common peppered moths because the actual phenotypic variation, that is, the variation between lighter and darker colored moths, existed in both pre-industrial collections and post-industrial collections. So in other words, there were two color forms of the moth. It just happened to be that the colored forms that were darker predominated after the Industrial Revolution, and the lighter forms predominated earlier. Well, Kettlewell thought about this, and he tried to come up with a logical reason as to what was actually causing this distinction in coloration. So here's a visual reinforcing what we already mentioned on the previous slide. Kettlewell's looking at these moths. He sees in earlier generations, for example, generation 0 over there, the lighter colored morph predominates, the darker colored morph is a minority. Then in the later generations after the Industrial Revolution, the darker colored morph predominates and the lighter colored morph is a lot less frequent. The big difference between the two is that Kettlewell reasoned no pollution in the early days and lots of pollution in the later days. As a matter of fact, it is a documented thing that the trees in England became coated, literally coated with coal soot because of the burning of coal, and those trees adopted this darker color because they had this layer of junk on them. As a consequence, you see that a darker colored moth, if it was to land on a tree, would be easy pickings if that was before the Industrial Revolution. It would stick out like a sore thumb. A bird flying by would see the black moth, eat the black moth, i.e., black moth has no fitness before the Industrial Revolution. On the other hand, you get all the soot, you got the pollution, the black moth lands on the tree, it blends in, it avoids predation, bumps up the fitness. On the other hand, the lighter colored moth, who's living life free and easy in the pre-industrial revolution era, in the post-industrial revolution era, lands on the tree, is a easy meal item for a bird, and as a consequence, the lighter colored moths lose their fitness. Hence, the shift in genotype, hence the shift in phenotype as illustrated in this picture here. Now, this turns out to be one of those sad stories of biology because there were plenty of scientists back in the 1950s who viewed Kettlewell's conclusions with a lot of criticism. And the criticism was so intense that Kettlewell just basically stopped doing any kind of research in this way, shape, or form, and died only a few years later. Ostensibly kind of an unhappy guy. And that's too bad. But the fact of the matter is in the years after his death, as we moved into the 1960s and to the 1970s, there were data that were collected that essentially supported Kettlewell's conclusions overall. Let's take a look at this graph. In 1960, smoke pollution was pretty high in the United Kingdom. Through pollution controls and improved awareness of the environment, smoke pollution levels dropped. And as smoke pollution levels dropped, one would assume there would be a shift back towards the lighter colored moths in terms of their fitness. In other words, as the smoke pollution decreased, the trees got cleaner. As the trees got cleaner, the darker colored moths didn't fare so well. So I see a second line on this graph. It is labeled number of melanic moths, and I notice that the number of melanic moths are decreasing through the 60s, through the 70s, through the 80s, right in line with what Kettlewell's original prediction was. So to put kind of a happy face on what amounts to being a sad story, even though he was dead, Kettlewell was justified in the end. Now, I just want to make one more comment with respect to the common peppered moth example here. This is an example of microevolution. You'll remember that microevolution is change within a species. So we're starting off with a common peppered moth, and we're ending with a common peppered moth. The only thing that's changing between time 0 and time now is the genetic composition of the population and the phenotypes of those moths. We have been focusing on natural selection as the mechanism of evolution. To be accurate, natural selection, while it is the primary mechanism of evolution, is not the only mechanism of evolution. See, evolution is change. Genotypic and phenotypic change in a population, anything that triggers phenotypic change or genotypic change in a population qualifies as a mechanism of evolution. So we are going to identify 4 different mechanisms of evolution in addition to natural selection. And as we cover these 4 different mechanisms, we'll try to remember that natural selection is the overriding mechanism that drives the process. So here's our list, and this is where we're going. We mentioned natural selection, the primary mechanism of evolution that in turn acts on all others. We will have a slide on mutation, a slide on gene flow, a slide on non-random mating, and a slide on genetic drift. And you will definitely want to get a handle on each of these 4, as well as being completely and totally certain on what natural selection is all about. We already mentioned mutation as the source of all new genes. And the fact of the matter is mutation is any change in a gene sequence that will ultimately create a change in a protein, a modification of protein. And sometimes the change in the modification in that protein might be to the organism's benefit, might be to the organism's detriment, or might not have any effect at all. So any spontaneous change in a gene sequence or a chromosome is going to affect the protein being made. The results could be lethal, detriment, benign, having no effect at all, or advantageous, having a benefit to the fitness of the organism. Now, not too long ago, you remember when we were talking about Hardy-Weinberg equilibrium, we said that populations in nature, as a rule, are open. An open population is a population that will permit immigration of individuals and new genes into the population, but consequently, you will also lose individuals or potentially lose genes from the population as well. That's what gene flow is all about. It is a characteristic of all natural populations. Immigration is the idea you can have new individuals coming in. Emigration, present individuals going out. And anytime you have an individual entering or leaving, you will have alleles or genes entering or leaving. Now, arguably, the fact of the matter is it's the alleles coming in that are going to have the greater effect on your population. So hence, looking at our picture here, I've got a population of brown beetles, got a population of green beetles, and I see a brown beetle is leaving its brown beetle population. Now, the fact of the matter is, as that brown beetle leaves its brown beetle population, it's probably not having a tremendous effect on the collective genotypes within that population. But you watch that beetle enter the green population, and all of a sudden you're introducing new genes into the green population, which could ultimately have an effect on the evolutionary trajectory of that population. Now, when I use the expression evolutionary trajectory, what I'm thinking about is what's going to happen to that population over time. If I've got a brown beetle entering that population and bringing some brown beetle genes with it, there's a very, very good chance that that green beetle population in the future might demonstrate some brown colored individuals. It all depends ultimately on whether those individuals are selected for. So once again, it's the fact of the matter is the gene is not going to become established unless it's selected for. So gene flow is a way I can get new genes into a population, but that's not going to matter a hill of beans unless that particular gene is selected for. At first glance, any idea of mating doesn't seem to have any effect or would not have any influence on evolution of a population. But let's take a look at what non-random mating is. You think from the standpoint of evolution, we've got individuals mating, exchanging genes, passing those genes on to future generations. And if the process of mating is completely, totally random, there shouldn't be any difference between the genes that exist at one particular generation and the genes that exist at another generation, because the choices of exchanging those genes is a completely and totally random process. But in non-random mating, you've got the reality of choice. Now, that means either the male of a mating pair or the female of a mating pair is going to select his or her mate on the basis of certain characteristics. We see this in the animal kingdom all the time. Now, in most vertebrate animal species, it's the females that exercise choice. They decide who they're going to mate with. And a lot of times that decision is made on some more or less overt characteristics of the male. How brightly colored is that animal? Does he do the right mating ritual? Are his horns really big? There's a number of different characteristics that fall into this category of non-random mating. But the fact of the matter is, as soon as that female chooses that male, she is preferentially selecting those genes, which means those genes will get passed on to future generations. Hence, if you are a male deer and you've got a big, huge set of antlers, a female might choose you as her mate, at which point those genes for those big, giant antlers and a number of other characteristics will get passed on to future generations. On the other hand, if you're a male deer and you look like Elliot, the deer in open season. I don't know if you guys are familiar with the movie Open Season, but the fact of the matter, Elliot is this idiot deer's only got one incy, teeny tiny little horn, and he himself is kind of a rather slight deer, and he doesn't have any redeeming qualities necessarily. But the fact of the matter is a female deer is going to choose Elliot. Those genes get passed on to future generations. But in reality, she's not going to choose Elliot. She's going to choose the big deer with the big giant horns. As a consequence, Elliot's genes will not get passed on to future generations. So you see what's happening here is the act of selection is occurring at the level of choice as to who you're going to mate with. So non-random mating is the fact of choice. And the fact of the matter is that if you select for these particular genes, like the genes in the big horned deer, the only way those genes ultimately get established in the population if they're selected for through natural selection over the long term. So just as in the case of gene flow, the selection for certain genes in non-random mating isn't going to mean a hill of beans unless it's acted upon by natural selection. So here's our fourth mechanism of evolution. It's called genetic drift. And genetic drift in a nutshell are the effects of random chance events on a population. So if you take a look at the illustration, the Amish woman on the right, she's holding a child, and that child is a dwarf. The child has polydactyly, which means he or she has, I believe in this case, 6 fingers on each hand and 6 toes on each foot. That's an example of a gene that could become prevalent in a population due to a random chance event. Now let's think about this. It could be a mutation that results in that dwarfism and in that event of polydactyly demonstrated in that baby. Now, the Amish are a very tight-knit group of folks. They emigrated from Europe to the United States about somewhere in the 18th century. And once they emigrated, it was only a very few families that actually emigrated to the United States from Europe. And so once they immigrated into our country, they set up their own little communities and they intermarried. And as a rule, what genes came into the country remained in those Amish communities. This is what is commonly referred to as a founder effect. So you had a founding number of individuals bringing these unique genes into their population, which admittedly remained small. Now, the fact of the matter is any genetic change in a population is going to have a greater impact on that population if it's small. So you think about it, if I have a community of 200 Amish people, and I've got these genes bouncing around that population, and it's not terribly large, just a few individuals inheriting that gene is going to have a substantial impact on the population on the whole. On the other hand, if our Amish population was several million individuals, might not have that much effect. Think about it another way. Suppose you've got a population of 100 birds living on an island, and those birds tend to nest in groups of 10. So you've got nesting groups of birds in groups of 10, which means in this population of 100 individuals, I've got 10 nesting groups of birds. Now let's suppose in that population of 100 birds, 10 of those birds have orange feet. The rest of them have blue feet. And it's one of these unique things that the orange-footed birds tend to nest together. So I've got 10 nesting groups of birds. 9 of the nesting groups have blue feet. 1 of the nesting groups have orange feet. Along comes a really, really violent storm. And that violent storm blows up on this island, and I've got this big, huge, giant lightning bolt that comes out of the sky and smacks straight into my population of birds. Now, just so happens, and this is completely entirely random, this lightning bolt strikes the nesting group of orange-footed birds. Destroys the nesting group, annihilates those 10 orange-footed birds. Now understand, because of a random chance event, I have completely lost most orange-footed genes in that population. Now, if I increase the population number of birds to 100,000 or a million, that one lightning bolt nailing one group of 10 nesting orange-footed birds might not have a heck of a lot of effect on the overall population. So the thing to remember about genetic drift, it is the effect of random chance events. Random chance events could be acts of nature, could be an event of parasitism, it could be disease, it could be hunting by humans, it could be an event of colonization. But the bottom line is, once that does happen, it's going to have an impact on the future genetic trajectory of that population. And that's why we refer to it as the founder effect. Now, there's another consequence of this, and it is called the bottleneck effect. And I think about it this way. You take a look at the illustration at the very bottom of the slide. I see a bottle that's full of marbles, and that bottle represents a population, and there's a certain amount of genetic variation in that population. So let's suppose that's like a population of some animal in nature, and human beings are hunting that population in nature. Now, if we hunt that population to the point that there's very few individuals left, let's suppose it's cheetahs. There are only like 1,000 cheetahs left on the planet. Now, if I'm a conservation biologist, I might want to restore the cheetah population. But understand something, it's like turning that bottle upside down. What I got left in my cheetah population after an event of hunting, which admittedly is a random chance event, I don't have a heck of a lot of genetic diversity in my cheetah population. So I turn the bottle upside down. And what few marbles come out is what I am building my new cheetah population from, in the best interest of that cheetah population. Yes, I am trying to restore the cheetah population, but the fact of the matter is that random chance event of hunting has tremendously decreased the genetic variability in that population, and what happens to the cheetah population is going to be much different as if there was no hunting event to begin with. So the hunting event creates a genetic bottleneck, and that's why this is referred to as the bottleneck effect. Now, as is the case for non-random mating, as is the case for gene flow, and technically as is the case for mutation, none of these mechanisms that create change in a population are going to have any long-term effect unless natural selection acts on those genes after the event. Finally, our last slide in the presentation. We need to talk about modes of selection. This is going to be really nice reinforcement of what we've learned thus far, and it's going to help us to segue into our next presentation on speciation. Now, when selection takes place, you're selecting for or against certain genotypes, and technically, like I said, where the rubber hits the road, you're selecting for or against certain phenotypes, because it's the phenotype that ultimately determines whether this animal or this plant is going to be able to survive or reproduce. So I want you to spend some time in studying this particular slide, and definitely go online or definitely go into the book and find out more about these. We're going to identify what is directional selection, what is stabilizing selection, and what is disruptive selection. We're going to take a look at directional selection first. Now, the set of illustrations there show me that directional selection is the two illustrations found in the middle of the sequence. In directional selection, what I am doing is I am selecting for an extreme phenotype. So you take a look at the bell-shaped curve there, and let's say we're talking about body size. This could be a population of animals, and within that population of animals, I got The far majority of animals are in between, either big or small. But let's suppose I am going to select for larger animals. That's the area of the population that is illustrated with the little area of orange. If I select for those animals, that technically means I'm selecting against smaller sized animals. And if you take a look at the illustration at top, it's what I'm starting off with. But if you take a look at the illustration at bottom, it shows me what I wind up with. If I select for larger animals, you see the entire bell-shaped curve has shifted to the right. I'm selecting for an extreme of the phenotype, which means my animals, as time goes by, are going to get bigger. That's the genetic trajectory involved in directional selection. Now let's take a look at stabilizing selection. Stabilizing selection are the illustrations on the left. And in this case, I'm going to select for animals with the intermediate body size. Let's suppose really, really big animals are energetically inefficient and they don't survive and they don't reproduce. And smaller sized animals just don't have the body resources to be able to survive or reproduce either. So it just looks like animals smack dab in the middle have a better chance of surviving and reproducing. As a consequence, those genes that code for intermediate size are going to be selected for. So if I take a look at the illustration above, which is what I start off with, and the illustration below, I see that the bell-shaped curve has narrowed, which means the far majority of individuals in that population now demonstrate the intermediate body size because I'm selecting against the larger organisms and I'm selecting against the smaller ones. Now, probably the most critical one is the last example, which is disruptive selection. Now, in disruptive selection, what I'm doing is I'm selecting for both extremes of the bell-shaped curve. In this case, maybe I'm selecting both for the larger sized organisms. Maybe there's some characteristics of being big that improves your ability to survive and reproduce. But then maybe, on the other hand, there are some advantages to being small. Maybe you can run faster. Maybe you can hide. As a consequence, the fitness of smaller organisms and fitness of larger organisms are going to be selected for, whereas the intermediate size is not. And so what I see in comparing the graph at top, which is what I start off with, to the graph at the bottom, which I see what I wind up with, is I see what I would describe as a bimodal distribution of phenotypes. Got two peaks. One peak representing an increasing number of organisms that are smaller, and the other peak on the right indicating a number of organisms that are getting larger. Don't have as many organisms smack dab in between. Now, what we're doing is we're setting the stage here for divergence. Technically, in the long term, the group of organisms on the left, the smaller organisms, could at some point become so significantly different from the organisms on the right that they might not any longer be able to interbreed with one another. And so I'm setting the stage for what we would call speciation, the creation of new species. We're going to call this disruptive selection because it disrupts what's going on in the population over time. We're selecting for the two extremes of the population and could, in fact, set the stage for the genetic trajectory of that population so that ultimately I might get two species arising from one. And this will have application when we take a look at our next presentation.